1 second.
I think you can see my first slide, can't you?
Yes, and if I can just take a little bit of time to officially introduce you, we are just so grateful today to be able to have this webinar and to have invited all of you. I've titled this webinar with Dr. Marco Ruggiero, What is the Third Brain and How Can Its Discovery Help with Boosting Immunity? And then I, of course, said for our children, but it's really in regards to anybody. We have Carolyn Tweetmeyer on the call as well, my dear friend, mother of 15 children, 8 whom are adopted, 7 by birth. Myself, I am the proud mom of 7 children, 5 that have been adopted. So we just really represent some moms who have been advocating for health, especially for, just children all over the world. And Carolyn's nonprofit is really to help advocate for children with HIV and severe special needs, Carolyn, who are in need of homes. Is that yeah.
Homes, yeah.
And it's projecthopeful.org is the name of her organization. And she helped me, just as she trailblazed, really to be one of the first people that I ever knew that had adopted a child with HIV. She's helped to educate us. She helped to equip me and enable me so that I was able to do the same. And she's done some amazing work. And then we have the amazing Dr. Marco Ruggiero, who is presenting tonight. And really, we are just standing by with full excitement to really see this presentation. But some of you may not know who he is. Marco Ruggiero, was born in Italy. He holds a PhD in molecular biology, is a medical doctor specialized in clinical radiology, and has been a professor of molecular biology at the Department of Experimental and Clinical Biomedical Sciences of the University of, is it Florence, Italy, for more than 20 years.
Yes, it is Florence, Italy.
And he worked at Burroughs Wellcome in North Carolina. I don't know if you knew this, but I used to live in Cary, North Carolina. Publishing a seminal paper on signal transduction with Nobel laureate Sir John Vane, and subsequently at the National Cancer Institute of the NIH in Bethesda, USA, working with Dr. Stuart Aronson and Professor Peter Duisberg. He has published more than 150 peer-reviewed scientific papers on signal transduction, reporting original observations in the fields of neurosciences and cancer. In 1987, he published a paper on the molecular mechanisms underlying the processes of learning and memory in the prestigious European Molecular Biology Organization, Journal of the Nature Publishing Group. Telling you something, these are a lot of big words for a mom like myself. But ever since, he published several papers in those fields of neurosciences that are associated with autism. And then one such topic on the frontiers of human neuroscience associated with Nature's Publishing Group, which describes the neurological alterations in the brain of autistic children and the use of ultrasonography in the diagnosis of treatment of autism. In 2014, he has developed an integrated protocol for the treatment of autism spectrum disorders that led to significant successes, with some autistic individuals fully recovering from the disease and able to resume a completely normal life. Some of these clinical reports were described in popular press. So this is actually documented. And if you need to read his bio, you can find that at bravousa.com. That's where I got it. So I'm just going to hand it over to you, Dr. Ruggiero, because I know we have some exciting information to really take in.
Thank you, Diana. Thank you, Caroline, and I thank you for this wonderful introduction. And I begin right soon to slightly contradict you, or better, not to contradict you, but I will begin my presentation talking not about autism and neuroscience, but talking about HIV and AIDS. And then we see after the first half of this presentation if you want to go on or if you want to stop and maybe begin the question and answer session, and we can go and talk about autism another time. Before we start, a couple of disclaimers. First of all, the declaration of conflict of interest. I am the co-inventor, together with my lovely wife, Stefania Pacini, of a probiotic yogurt that is called Bravo, and I hold shares in the companies that produce and distribute this yogurt in Switzerland, Europe, and the USA. I'm also the inventor of a new product that you probably never heard of. It is called Rerum, and it is an immune stimulating factor that is based on chondroitin sulfate. It is an evolution of the old GC macrophage activating factor. And also, this disclaimer, I think it is important because none of the statements, observations, results that I will be showing have to be interpreted as medical prescription or advices. They are academic and scientific consideration that you are free to use as a starting point for discussion with your trusted healthcare professional if needed. And it is important to state that none of the statements has to be interpreted as a cure for any given disease. We are not curing autism, we are not curing HIV AIDS, we are not curing a cancer or any other disease. We are simply sharing observation that may help cope with a number of diseases or also help us to live healthier, longer lives. As you said, I began working in research in the early 80s when I was a young medical doctor. This is the very first paper I published on the relationship between food, nutrition, and cancer. It is in Italian, but you still can find it in the National Agricultural Library of the US Department of Agriculture. This is to say that I was interested in the role of nutrition in health and disease since the very beginning of my career. You mentioned the paper that I published with the Nobel laureate Sir John Vane when I was at Burroughs Wellcome in North Carolina. This is the paper I published in 1986 where we described the molecular mechanism of action of some molecules that later on, in particular the protease inhibitors, are used in the treatment of HIV infection and AIDS. You probably heard this name, protease inhibitors. This is where some of the research started in the mid-80s. Then I moved to the National Cancer Institute in Bethesda, Maryland. I was working on oncogenes, the genes causing cancer. And this paper, published together with my colleagues in the journal Science, eventually led to the development of this new type of a biological anti-cancer drug that is called Herceptin. It is marketed by Roche. I have no involvement with Roche at all, but this is where the research started and where this product was initially conceived. In the early 90s, in August 1990, I was interested in the role of the immune system in fighting diseases. In those days, working at the National Cancer Institute, of course, I was mainly concerned with cancer, and I published in the Proceedings of the National Academy of Science of the USA this paper describing how activation of macrophages could empower the immune system to fight cancer. 20 years later, I was presenting together with my wife and Dr. Yamamoto at the World Conference on AIDS that was held in 2010 in Vienna, Austria. This study that was accepted after peer review, where we elucidate the cellular and molecular mechanism through which a stimulation of the immune system leads to eradication of HIV. I know these are strong statements, but these are written in the most prestigious venue, that is the World AIDS Conference. Now, in 2011, together with Peter Duesberg and a number of other scientists, we published this paper, published in a rather minor journal, the Italian Journal of Anatomy and Embryology, that reviews the actual size of the epidemics of HIV and AIDS all over the world and in Africa, and shed lights on the importance of nutrition, or in the case of Africa, of malnutrition, in the onset and development of HIV infection and AIDS. And now let's go to the topic that it is of our interest. It was in 2010 when we were looking for natural methods to empower, to strengthen the immune system with the ultimate goal of fighting HIV infection and AIDS, and why not also empowering the immune system to fight cancer and other diseases, when we ran into this paper that was published in 2010 by a group of scientists coming from Canada and led by Dr. Gregor Reid, who is an authority in the field of microbiology. This scientist had run a very interesting experience in Tanzania, Africa, one of the poorest countries on this planet, where they taught local women to prepare a probiotic yogurt from which we took inspiration to develop our product. And so in Tanzania, in Mwanza, in an impoverished neighborhood of this part of the country, they set up a sort of a local production of yogurt, and these women, they gave the yogurt to HIV-infected people and AIDS patients. And the result was astonishing, and they published this in this Journal of Clinical Gastroenterology, and as the title says, they noticed an increase of CD4, the cells of the immune system that are targeted by the HIV infection, that are typically low in HIV-infected patients and in AIDS patients. So simply the probiotic yogurt consumption was associated with the increase of CD4. This was truly an astonishing result that led us to the development of a probiotic yogurt that was based not only on their observation, but also on our studies published in the same year, 2010, in Vienna, describing the molecular mechanism at the basis of the eradication of HIV infection. So it was then in 2011 when we took courage and we went to Rome, Italy, at the 6th International AIDS Society Conference on HIV pathogenesis, treatment and prevention. So this was the World Conference on AIDS that was held the year after the one in Vienna, and the focus was treatment and prevention of HIV infection and AIDS. And this paper is still, of course, online. You can go and retrieve it. Was accepted after peer review. And here we describe for the very first time this product that came out from our research. In those days, we were not marketing this product, and so the nickname was MAF, macrophage activating factor 314, because it took us 314 experiments before we could find the formula that truly was effective in reconstituting the immune system. Now, the next slides that I'm showing you and that you can still find in the website of the International AIDS Society show the actual results obtained in Italy. So you can see these are the actual print screens of the exams of two subjects, one of which was moderately immune depressed. Now, this subject number one, the count of the CD4s was below the threshold of 400, that is indicative of AIDS, of full-blown AIDS. At least in Italy, below 400 CD4s, you are no longer simply HIV infected, you have AIDS. So before consumption, this subject had 372 CD4 cells. And only after 8 weeks, 2 months of consumption of the yogurt with no other treatment, the CD4s doubled or more than doubled. They went up to 853, and so the subject could not any longer be classified as AIDS. Now, the other subject is even more interesting because this was a female, and the number of CD4s were fairly high, 857. The point is that the number of the CD8 that are so-called suppressor, those cells that inhibit, block the immune system, was too high. And therefore, the ratio CD4-CD8 was too low. In other words, this lady had a good number of good soldiers, the CD4s, fighting for the immune system, but also had a high number of those blocking the immune system. It's very interesting to notice that only after 3 weeks of consumption, not only the CD4s went up by a figure of about 400, but the CD8 went down. In other words, it was not simply a non-specific stimulation of the bone marrow. It was a rebalancing of the immune system, rising the number of the good soldiers and decreasing the numbers of those who tried to stop the good soldiers. So in this case, the ratio went up and went back to normal. And this happened only after 3 weeks. But these results by themselves don't tell you too much if you're not in the field of HIV and AIDS. Just to give you an idea, according to the literature, when you take the antiretroviral drugs, you expect an increase, on an average increase of about 90 cells in 1 year. 90 cells in 1 year. In this case, using only a nutritional approach based on this probiotic yogurt, we saw increases of 400 in only 2 months. So definitely a result that, by the way, was fully consistent to the results that Gregor Reid and colleagues had reported from their experience in Tanzania. We also wanted to look at other cells of the immune system, the so-called NK or natural killer cells. You go to Wikipedia and you find out that these cells are a type of cytotoxic lymphocytes that constitute a major component of the innate immune system. They are the cells that prevent the onset of tumors and that kill the cells infected by viruses. So they're extremely important in our immune response. Obviously, the subject number one, the one who was moderately immune depressed, had a low level, 165 of these NK cells, that went up to almost 400 in only 2 months of treatment. And also the other subject, who had a fairly decent level of NK cells, 251, saw an increase of about 90 cells in only 3 weeks. So from this data, that may seem a little bit dry, not too passionate, we can say that we were able to rebalance and stimulate in the most natural way the immune system within week, and quite obviously without any side effect. Now, these results were confirmed 1 year later in 2012 by the same group of scientists, you see Gregor Reid from Canada, when they observed the same identical results, identical to ours, identical to theirs obtained in Tanzania, this time in London, not London, UK, but London, Canada. This is to say that this is not something that works only in malnourished, impoverished areas like in Africa, where you could argue, well, fine, those are malnourished people, simply nourish them and they will get better. Not at all. In Italy and in London, Canada, we have not malnutrition. On the contrary, if anything, we have overweight. And nevertheless, the effects of these probiotic yogurts, they are very, very significant also in those parts of the world where malnutrition is not an issue. Now, this is one of the many emails that I received. This was received today at 1:40 a.m. I don't know what time it was when the guy wrote it. This guy is a lawyer whose son is HIV positive. Now, as it says here, Peter, who is HIV positive, tried the Bravo, and again, the viral load, that is the number of viral particles in your bloodstream to make it short, went down in only 2 months. So again, 8 weeks, maybe 9 weeks here, from 12,000 to 6,000. And the CD4 cells that the lawyer calls them T cells went up in the 60s after having been in the 600 in the 400. So again, we see a consistent increase of CD4s of about 200 cells in about 2 months. But let's move on to some other ways of administration that we have developed in these years. Of course, a yogurt can be drunk, but also there are other ways to reconstitute the microbiome, that is the gut microflora, and together with pharmaceutical company in Nuremberg, Germany, and in Switzerland as well, we have developed suppositories that are made with yogurt, with live cultures. If you put one of these suppositories in milk, the next day you will have a yogurt, because it was a kind of a difficult technique, but eventually we succeeded. And the next slides that I want to show you demonstrate that this approach of ours doesn't work only with HIV infection. Since its goal it is to empower and strengthen the immune system, it works against all types of viral infections. This Mr. Zunaid, I don't know whether this is the first or the last name, so I don't know whether I erased the right name, whatever, is a doctor actually, who is infected with the hepatitis B virus. Before he started treatment with suppositories, he lives in Thailand, as you can see from these characters, his viral load was in the range of the 5 millions. So 5 millions of particles before he started the treatment with the suppositories. Now, about 1 month later, that is in October of 2014, his viral load went down by 90% because from 5 millions, now it was 284,000. A few months later, the viral load went down to 20,000. So we see a 99% elimination of the hepatitis B virus within a few months by simply reconstituting the human microbiome, in this case through suppositories, because we know that the human microbiome is the immune system. So these results tell us that our nutritional approach is not valid uniquely against HIV or AIDS, but also against all types of viral infections. And as we will see later, by rebalancing, re-establishing a correct function of the immune system, it also works in other diseases. Oh, I forgot, is a viral load went farther down to 12,000. So now it's difficult to say it is a 99.99%. His viral load was almost undetectable. Now, you may say, fine, we stimulate and we rebalance the immune system. But there are people who have autoimmune diseases like lupus or psoriasis or pemphigus, which is a disease mainly dermatological, and we don't want to stimulate the immune system in these people. Actually, these people, they take steroids or immune suppressant drugs because their immune system is functioning too much. Well, when you re-establish and you rebalance the immune system in a natural way, you expect good things to happen. Now, the next slides are not for the weak of heart, for the faint of heart, but we're talking about medicine, so be prepared. They are not pleasant, but the end of the story is very pleasant. Now, the following are the lesions in the foot and ankle of a patient having pemphigus. Pemphigus is an autoimmune disease of the skin. And this is also another picture of the ankle and the leg. Now, this patient, who is patient of a doctor who works in California, in San Diego, took the yogurt, and after about 2 months, no other treatment was involved. These are the results. So you can see what is called baby skin. All the lesions are almost gone, they are peeling off, and you see that the skin is being regenerated in the most natural way. So you can see truly the baby skin, and the old dead skin now is peeling away, and so this patient is completely recovering from his condition. But now comes a part of the story that will leave you without breath, at least left me without breath. Because so far I've not shown things that are too revolutionary. Because there is a general agreement, because of what we know of the microbiome, that the consumption of probiotic yogurts leads or may lead to the increase of CD4 cells in HIV-infected people, may lead to the normalization of the function of the immune system, and therefore to the improvement of the symptoms associated with HIV infection rates like diarrhea, weight loss, and so on. However, the following paper will show something that was completely unexpected and most likely even escaped the attention of the authors themselves. Be prepared, because here there will not be strong images, but there will be very strong information. Now, the paper starts with something very easy. Same research group led by Professor Dr. Gregor Reid from Canada, who work in Mwanza, Tanzania. What do they do in this paper? Something very simple. In 2011, that is 1 year after they had observed that the consumption of the yogurt led to an increase of the CD4s in HIV/AIDS people and an improvement of the symptoms, they went farther ahead and they reasoned, why don't we give them probiotics instead of the yogurt? Yogurt is rather complicated to be produced, so why don't we simply encapsulate these probiotics and let's see what happens to a selected group of women. Don't ask me why they chose women and not male, maybe because they were more under control. I don't know. So essentially, they collaborated with this treatment clinic at the Seoko Turi Regional Hospital in Mwanza, Tanzania. And of course, you had to be confirmed HIV infection to participate in the trial. So the trial essentially was designed that HIV-infected women, not yet full-blown AIDS, and therefore not yet eligible to be treated with antiretroviral, took the probiotics. And then they say, let's see what happens to the CD4s and all the other things. So all these women were screened for HIV, and obviously only those with confirmed HIV infection were enrolled in the study. And that's something that they didn't expect happen. In the women assigned to the probiotic group, there were 33 women assigned to the placebo group and 32 assigned to the probiotic. 2 of them became HIV negative. Of course, nobody is saying here that probiotic yogurts make you HIV negative, but the observation is here. It is strong. It is published. So women who were confirmed HIV positive, maybe it was just a random coincidence, who knows? The point is that only 2 of those assigned to the probiotic group became HIV negative. Now, these events are rare, but actually there is ample evidence in the peer-reviewed literature that HIV positive subjects may revert to HIV negativity, very often after a strong stimulation of the immune system. The following case came to the attention of a good friend of mine, Dr. Klaus Köhnlein, working in Kiel, Germany, and essentially my role was simply to evaluate the results and to write the paper. The paper has not been published as yet, but I have permission to show the results. Now, this guy is a guy living in Germany of Italian origin. I don't know if you can see here on the right. Let me move this. Okay. Here on the right, this guy was diagnosed HIV positive with all the high-tech tests that they routinely do in Germany. So this guy was HIV positive, and he was positive to all the proteins of the HIV as usual. Now, oh, why it does not go? Okay. Now, this guy also had a high viral count, 85,000 copies of the virus in his blood. Now, this guy, just by coincidence, developed a foot infection. Fuß in German means foot. Erysipelas is an infection by Staphylococcus and Streptococcus. And this infection, of course, leads to a very strong stimulation of the immune system. 5 months later, he went back to Dr. Kuhnlein for a further assessment. In the meantime, he had done no treatment at all, and these are the results. HIV negative. And also the Germans usually are very cool. They put an exclamation point because they couldn't realize how is it possible that this guy was confirmed HIV with a high number of viral copies in his bloodstream has become HIV negative. Well, nobody knows, but most likely the strong stimulation of the immune system coincident with the infection, with the bacterial infection in his food probably had led to the elimination of the virus by the immune system in itself without the help, in this case, of any drug. Now, the guy speaks in Italian, and as you can see, the previous slide, when he became HIV negative, was dated in May of 2011, and just a few days ago, that is in November, well, more than 1 year ago, in November 2014, he wrote me an email. The guy works in the pharmaceutical industry, so there is a kind of irony. And he says, since I work in radiochemistry and I am so-called at risk because of the work with toxic elements and so on, I have exams every 6 weeks and all the values, they are normal. So not only he became HIV negative, but 3 or 4 years later, he is still in excellent shape. Now, this is the title of the paper that we want to publish in the British Medical Infectious Diseases, has not been published as yet, where we describe the spontaneous regression of HIV infection in a 42-year-old patient, a case report. But we are not the only one to have observed this phenomenon. If you go to PubMed, you find this paper that was published in a Chinese journal, and it is only available in Chinese except for the abstract that is written in English, where they do report 8 seronegative converted HIV/AIDS patients with traditional Chinese medicine. So 8 confirmed HIV/AIDS patients turned negative after, in their case, treatments with traditional Chinese medicine. I don't know the details of these treatments, but I wouldn't be surprised if these treatments naturally empowered or stimulated the immune system. Now, as you know, the HIV virus was discovered by the French professor Luc Montagnier, and he stated many years ago, we can be exposed to HIV many times without being chronically infected. Our immune system will get rid of the virus within a few weeks. And we have seen this happening. But only, I add, if you have a good immune system. And in Africa, or also in North America and in Europe, a good system cannot work properly if yet it has not the appropriate nutrition. And this is one of the major problems both in the developing and in the developed world, the adequate nutrition to empower the immune system. Now, there are several ways to empower the immune system through nutrition, but this is one of the most accepted views, that is to limit the consumption of carbohydrates and to prefer instead monounsaturated and saturated fats like extra virgin olive oil or coconut oil with anti-inflammatory properties and a good amount of proteins. Now, talking about proteins, we made some interesting observations in the recent past, because unfortunately, given our lifestyle and given also the availability of proteins, for example, in the third world or in other parts of the planet, it is not always easy to have the best proteins available. So there are many products, and I'm sure that you know a few of them. So here I took care to remove any brand name, but there are several proteins, several products that can be prepared like shakes. Shakes that are essentially made with milk proteins, whey protein concentrate, and milk protein concentrate. Now, of these shakes, you can find hundreds, if not thousands, on the shelves of grocery stores and specialty stores. But there are a few of them that are peculiar, like this one that I have found, because not only they do contain the proteins that essentially come from dry milk, but they also contain enzymes. You remember the work I did with Sir John Vane? Proteases. Proteases are enzymes that clip, digest the proteins. And this particular formulation also contains lipases, that are enzymes that digest the lipids, and also enzymes that digest the carbohydrates. So in other words, this is a very interesting formulation because it contains the proteins, the enzymes to digest them, and also it contains vitamins. No problem, there are many vitamin supplements on the market, but also minerals. Why this combination is so interesting? Because since we know that the enzymes need time and temperature to work, if you let incubate these shakes for an appropriate length of time, that is between 30 and 45 minutes at room temperature. So essentially you go against the instructions provided by the manufacturer, because you dissolve the protein shakes in water, you keep them on the table of your kitchen for 30-45 minutes at room temperature with no ice. What happens? It happens that essentially the enzymes, they pre-digest the proteins, the lipids, and the carbohydrates. So what you will be drinking after 30-45 minutes has nothing to do with the stuff that you have dissolved. It is now something completely different, because from the proteins, now the proteases have generated peptides that have powerful biological functions, because they do stimulate the immune system, they have anti-cancer properties, they have properties that favor the circulatory system and the heart, and so on. So if you simply are patient enough to wait 30-45 minutes, you will have something that not only is an excellent nutritional support, you have something that exerts a number of beneficial healthy effects. Now, if you take the yogurt and you mix the yogurt together with this shake or other shakes that have a similar formula, now the enzymes, they work together with the yogurt, and at the end you have something even more powerful. But let's be wary, because proteins alone are not sufficient. In other words, if you want to see truly a good stimulation or a rebalancing of the immune system, an immune system that can successfully fight viral infections and cancer, you also need other nutrients, other macromolecules. That's why some products are not so efficient. They're good nutritional support, but they don't do much more, because they lack a fundamental molecule, macromolecule, that is called chondroitin sulfate. Now, chondroitin sulfate, whose chemical formula is reported here, again, this comes from Wikipedia, so it's freely downloadable. It's a very interesting compound. You go back to PubMed, and in 1998, it was demonstrated that chondroitin sulfate is a potent inhibitor of HIV. But even most interesting is that chondroitin sulfate reduced the viral load, that is the number of viral particles, to non-detectable levels in 10 days. I'm not wrong, I'm reading right, 10 days. A result that obviously cannot even be imagined with the most powerful antiretroviral drugs we have today. So in 10 days, the chondroitin sulfate was able to reduce the viral load of HIV-infected patients to non-detectable levels. And then again, the Italians come to the rescue, because it was in 1999 when an Italian researcher, a certain Di Caro, reported that chondroitin sulfate not only inhibited the HIV, but it empowered the immune system up to the point that also these so-called opportunistic infections that are unfortunately associated with HIV and AIDS were inhibited, were counteracted by the chondroitin sulfate. So the chondroitin sulfate, this macromolecule that is present in the Bravo yogurt, and also it is formed during the incubation period of those shakes, because chondroitin sulfate essentially is part of milk. Now the chondroitin sulfate inhibited not only HIV, but also the herpes simplex virus type 1 and the human cytomegalovirus, both agents that are responsible for the opportunistic infections in HIV-infected people. And these authors concluded that in view of the absence of the side effects, a combination of these derivatives could have potential therapeutic effects. This is the paper that you find in PubMed, where they say that a combination of these derivatives may have therapeutic potential. Then don't ask me why. All this research that was published in the mid-late 90s was never followed up. Fortunately, we are following it today, and the products that I've shown you thus far, they are based on this research that now is almost 20 years old, and that has always been confirmed, never disproved. The conclusions of this first part of my talk, that could be also the last, if you wish, and we can go to the question and answers, is probiotic yogurts that reconstitute the microbiome and therefore the immune system, because we now know that the microbiome is the immune system, may help fighting viral infections. And we have seen several examples of these, from Tanzania to Canada, from Italy to San Diego, California, from Germany to other places in the world. Now, another point is that the proteins that have been partially digested in what we call in vitro incubation, that may simply happen on the table of your kitchen, may help us strengthening the immune system, because thanks to the presence of the proteases, during these 30-45 minutes, a number of peptides and other molecules like chondroitin sulfate are formed, and these molecules, they not only have a nutritional value, they are very powerful in rebalancing and stimulating the immune system. And when you combine this with the yogurt, of course, you have the best of both worlds, because you have the live microbes, but you also have all these molecules. And last point is that glycosaminoglycans such as chondroitin sulfate appear as promising agents that strengthen the immune system and help fighting viruses, plural, because as we have seen, all this is not simply related to HIV infection and AIDS. Since it is related to empowering the immune system, of course, whether you have an infection from hepatitis B or from Lyme, it doesn't matter, because the immune system is the one that takes care of all these types of infection. Now, I'll be speaking for 40 minutes, and I stop it here for a few seconds, and I let Mrs. Diana or Mrs. Caroline to decide together with the audience what we want to do. I am available to go ahead for another 20-25 minutes and talk about autism, or I can stop it here and we can discuss at length, and we can talk about autism another time. I'm here at your disposal, at your service, so please let me know how do you like to proceed.
Well, this is, I mean, I'm speechless. This is amazing stuff. And I know that Carolyn, I think that you had a couple things to say, just because you've tried this protocol, and we wanted you to just kind of give a little bit of a testimony, and then we'll maybe ask some questions.
Yeah, I mean, I would like to touch a little bit on the autism aspect if we can, just maybe for a few minutes, and maybe segue into our next get together. But I definitely would like to share a little bit about what has happened in my own house. You know, a lot of you know one of my daughters that has struggled and actually has an AIDS diagnosis, and her situation for a long time has been an extremely low CD4 count, and it dropped again recently before we started with the yogurt protocol. And what took us years in the past to slowly build her up to. We had a pretty extreme jump for her. Actually, it never happened before in this 8 years we've had her, you know, in the US using her ARVs. Within 3 months, she had an extraordinary jump that we hadn't seen ever in the history of her being here and following just the regular medical protocol. So on top of what we already were doing with her medication, we simply added this, and we can't deny that some pretty awesome things have happened as a result. And on top of that, our house is like a giant petri dish. We've dealt with some other significant issues. We've got an H. pylori circumstance that we have seen incredible things happen with. We have a son with autism that, you know, there's some undeniable things happening here that weren't happening before we started. And our kids with Down syndrome as well. Just some incredible things are going on. I can't explain it, but I can't deny it. So I just want to share it, and we're just so excited to share the potentials and possibilities of something that is just so, which it sounds so simple. You know, basically just hearing that this is essentially yogurt. That is obviously so much more, but it is doing things. It is absolutely doing things. And again, we're so thankful to you to give us this time and to spend with these mothers that are just searching and searching for answers for their children. And I know they've gone across the world to get these kids and bring them here, and everything is so far beyond the call of duty that to deliver this to their living rooms at this point in time is such an incredible gift.
Yeah, and I'll tell you that the consensus, if you don't mind, is that they would love to hear you talk about autism. There was one question, if you don't mind just clarifying, was the room, it's all written here, but if there's any more that you could add to the room temperature aspect of the enzymes, you know, developing and sitting for a while. I don't know if there's anything else to elaborate on, but there was some confusion, and because I'm following some texts in the thread.
Well, on the room temperature, the concept is very, very simple. Some of the shakes, like the one that I showed before, they contain not only proteins and other nutrients, but they also contain the enzymes that are the same that we have in our body to digest them. Of course, if you pre-digest something, you already have the products of digestion that by themselves exert a number of healthy effects. So essentially, the instructions to obtain these healthy effects is to dissolve these shakes in water as usual, without ice, in water at room temperature, and then let them sit at room temperature in your kitchen or wherever you like in your house for 30 or 45 minutes. So you give the time to the enzymes to work, and the temperature. They don't like cold temperatures. So that's why you don't have to add ice. Then after 30 or 45 minutes, you have produced, not you, the proteases and all the other enzymes have produced a number of molecules that have a very positive effects on the immune system and on a number of other functions of our body. At that point, after 45 minutes, you simply mix them again. If at this point you want to add ice because you like ice-cold beverages, fine, you can do it now. Now, you can also add the yogurt that you had previously prepared at the beginning of those 30-45 minutes. In this way, you will add the properties of the yogurt. The enzymes will work now both on the whey and dry milk proteins that are in the shakes and on the proteins that are in the yogurt. So essentially, there is a synergism, and you increase both the potency of the shakes and the potency of the yogurts. So from the practical point of view, is very simple. Simply leave the shakes there for 30-45 minutes, or leave the shakes and the yogurt there for 30 or 45 minutes together, of course. Mix them well and enjoy them. And they're also very pleasant to the taste.
Can I ask you if you have any other suggestions overall diet-wise I'm sure people can't be eating Skittles and pizza and then just jump on this once a day and think everything's going to be great.
No. Well, why don't we do this? Why don't we plan a webinar talking only about nutrition? Because we have developed, and we are not the only one, specific nutrition for cancer, for autism, and so on. But it takes more than a couple of minutes. So let's go in detail in a future webinar about nutrition. But again, it's not something you can do once in a while. If you want the nutrition to work, you have to do it every day.
Right.
All right, so do we want to talk a little bit about autism?
Yes.
Now, please stop me when I am becoming too boring. I'll try to be as fast as I can, but still I prefer to be clear and to give less information, but clear rather than a number of things. Also because I hope that this is not the first and last time that we meet. So next time maybe we can cover other aspects that today maybe are not covered. Now, talking about autism, I will not again say that I have been working in the field of neuroscience for 30-some years. That's very boring. So let's see what we have done in the past few months. Together with the late Dr. Jeff Bradstreet, who tragically died in June of last year and who was an authority on autism and with whom we had published the papers on the pathogenesis, that is the cause of autism and how to approach autism. On the 22nd of December, so just a few days ago, we published in this prestigious journal that is called Frontiers in Neuroscience, a paper, rather short paper, about 1,000 words, that even though it is short, has received about 10,000 views. That is enormous for a scientific paper, and it has an impact factor that is among the highest in the fields of neuroscience. What is written in this paper? In this paper, we put forward an hypothesis that explains the lesions in the brain of autistic children, lesions that we, together with many other scientists, had described using different techniques like MRI or ultrasonography, and most important, how to address these lesions. This paper derives from another paper that had been published by scientists in Nature just a few months earlier, where they describe lymphatic vessels in the brain that had never been described before. Before this paper published by these researchers, it was thought that the brain was, as we say, an immune privileged organ. That is an organ outside of the immune system, not connected with the general immune system. Now, this paper proves that this conception was wrong. And as they say, one of the characteristics of the central nervous system is, or was, the lack of a classical lymphatic drainage system. So now this paper describes that actually there are lymph vessels in the brain. And as they say, the discovery, this is not us, these are the other scientists, the discovery of the central nervous lymphatic system may call for a reassessment of basic assumption of neuroimmunology and sheds a new light on the etiology of neuroinflammatory and neurodegenerative diseases associated with immune system dysfunction. So I don't know why it didn't come in to their mind that also autism could be related to all this. So our contribution was to think, why not? Also autism can be caused by some dysfunction of the immune system inside the brain through the discovery of this lymphatic system. Now, the lymphatic system of the brain drains, that is the lymph that is collected in the brain goes somewhere, and it goes into the lymph nodes that are inside our neck, essentially around our throat, around the jugular vein. Here, this is an old anatomical sketch, and these are the lymph nodes where the lymph coming from the brain arrives. Now, the lymph to these lymph nodes also come from the mouth and the throat, and I think you begin to realize what the problem could be. Now, this is us, Bradstreet, Pacini, and myself. And we ask ourselves a rhetorical question in this paper published in Frontier. Could inflammation-associated deficit in meningeal lymph drainage be the culprit for the observed increase of extraaxial fluid, that is inflammation in the brain? In other words, we postulated, if we develop an infection in the mouth or in the throat, then the lymph nodes inside the neck become swollen, become clogged, and because of this, the lymph from the brain cannot drain appropriately. And so there is an accumulation of lymph inside the brain. With the accumulation of liquid, now the neurons and the glial cells cannot connect with each other, because the brain, it is inside a closed cavity, that is the skull, cannot expand, and therefore, if it is an accumulation of fluid, due to the inflammation of the lymph nodes in the neck, due to some infectious process in the mouth or in the throat that is very common, or in the nose, then the lymph remains in the brain, and let's see what happens. As we write here, infection or inflammation of the deep cervical nodes that may be due to infection or inflammation in the mouth or in the throat or in the nose may lead to impaired lymph drainage, with consequent accumulation of fluid, inflammatory fluid in the brain, and this leads to the disruption of the connections between neurons and glial cells, exactly something that we have published in 2014, again in Frontiers in Human Neuroscience. These lesions that we can see in the brains of autistic children. But there is much more. Now, if the lymph from the brain cannot flow and cannot be eliminated, then of course you have accumulation of the metabolites, the exhaust fumes of the metabolism of the brain. If you want to call them toxins, you are free to do it. And this leads to constant inflammation of the brain and the meninges. And if you are in the age of development, this leads to alterations of brain development and function, that is autism. If you are old, this leads to all types of neurodegenerative, neuroinflammatory diseases such as multiple sclerosis, amyotrophic lateral sclerosis, Parkinson, and Alzheimer. There is something even more interesting. Now, if the lymphatic drainage, that again is the flow of the lymph from the brain down to the immune system, is impaired, of course, the immunological defenses of the brain are decreased, and it is decreased the capability of the brain to fight pathogenic microbes, microbes that penetrate into the brain from the intestine. Now here I borrowed a slide from the famous French philosopher Jacques Derrida, who obviously in French wrote about himself, I must be crazy to write this. And I know that I sound very crazy in saying in public and even encouraging you to share this, that there are pathogenic microbes that penetrate into the brain from the intestine. Now, as you know, and we've been talking for about 40 minutes, I have some experience in the field of HIV and AIDS. So let's go back to the field of HIV and AIDS, and let's look at this paper that was published in PLOS by these researchers, again coming from Canada. What did they do? Essentially, their study began like a simple study. They were interested in assessing whether in the brain of people who had died of AIDS, they could find microbes that were indicative of opportunistic infections. So again, something rather trivial. You have AIDS, you have opportunistic infections, it is likely that after you have died of AIDS, we find microbes in your brain. The point, however, came when they began looking at the controls. They were doing autopsies. So controls are people who have died of a heart attack or a cancer, but definitely have no immune deficiency, have no HIV or AIDS. Now, they found in the brains of the controls a number of microbes, without any sign of infection or any sign of inflammation. And they were forced to conclude something that defies, or it is at the verge of science fiction. So let's read together with them. They begin very easy. The existence of commensal microbes that colonize organs within the human body has long been recognized and termed microbiome. So we all know that we are not sterile. We have a gut microflora. We have microbes in our nose, throat, mouth, on our skin, and all these microbes are called microbiome. However, until this paper, it was thought that the brain was sterile. Of course, unless you had some serious encephalitis or meningitis that are very serious disease. And they write, in an organ, the brain, widely assumed to be free of infection agents in the absence of a specific disease process. In other words, they did not autopsy the brains of people who had died of encephalitis or meningitis. They were people died, let's say, of a heart attack. They found a number of microbes. Microbes that are the same that you find in soil and water. In other words, our brain is not sterile at all. There are the same microbes that you find in soil and water, and obviously the same microbes that you find in your intestine. How do these microbes go to the brain? They go because they are carried by cells of the immune system and our good friend, the macrophages. Macrophages that have been the object of my research since 1990 when I published that paper in PNS. So in other words, the macrophages, they pick up the microbes from the gut and elsewhere, and they carry them to the brain through the lymphatic vessels that had not been discovered until a few months ago, and that Brassett and myself highlighted their importance. So, summarizing. In the brain, there are microbes that are commonly found in soil and water. The cells of the immune system, mainly the macrophages, carry these microbes to the brain. And we hypothesize that the brain lymphatic system through which the immune cells travel is instrumental, it's like the highway, to carry the good or the bad microbes to the brain and influencing its function. Of course, do you think that if there are millions, billions, or zillions of microbes in your brain, they're doing nothing? Obviously not. As the researchers who published in PLOS write, the capacity of microbes for influencing brain function is immense. For a scientist, it's not easy to say it is immense. It means it's beyond recognition. Obviously, now that we know that microbes are part of our brains, don't think that they're doing nothing. They influence the function of our brain just like neurons and glial cells do. So now we have to deal with a totally new concept, the brain microbiome, the microbiome inside the brain. In autism, the role of the gut microbiome was widely recognized, and there are thousands of papers describing the gut microbiome as a new frontier in autism research. And we know that experimental changes in the gut microbiome can affect emotional behavior. All these are papers published in PubMed. And microbes manipulate the host, that is our eating behavior, to increase their fitness. And microbes are directly implicated for the development of psychobiotic, that's a new word, not anymore probiotic, but psychobiotic, that is microbes that influence the way we think, our depression, our mood, voices from within, gut microbes and CNS. All this was known, but until now, including ourselves, when we presented in 2015, a few months ago at Autism 1, we thought that the brain, the immune system, the gut, and the microbiome were not separated organs, but different anatomical and functional components of the same organ. And therefore, we developed this comprehensive strategy to target all these components at the same time. But we still were wrong, because we thought that the microbiome in the gut influenced the function in our brain. And so the reasoning was, let's fix the gut, and this will reflect on the brain. We were wrong. The microbes are inside the brain. And in the past 5 months, we have learned that the interconnection is much stricter than what we had imagined. The microbes are cells of the brain, just like in the neurons and the glial cells. The microbes are as important as the neurons and the glial cells. The microbes arrive to the brain, to the immune system. Now we understand why immune dysfunction is related to neurological issues, because the immune system carries the microbes. And this could sound like a spot. The microbes that you have in the intestine are the microbes that you have in the brain. And probably you want to have good microbes in your brain. And if you want to have good microbes in your brain, you want to have good microbes in your gut. In addition to this, you want to have a functioning immune system inside your brain, and you want to have a functional lymphatic drainage so that the lymph can flow from the brain down to the immune system. That's why we developed this protocol that today I will only touch briefly, and of course I'm available to expand on the diet, ketogenic diet, the reconstitution of the brain microbiome, and the targeting of these deep cervical nodes. So I will go rather fast, but I'm available to expand and elaborate on these points in another webinar. Now, until recently, and please, Diana and Caroline, stop me whenever you feel, looks like you are masochistic, you want to hurt yourself listening to me. Okay, until you switch off the connection, and then I realize that I say too much, I keep on talking. Now, until recently, the main target, as far as reconstituting the microbiome with a probiotic yogurt, was the gut microbiome. And we knew, as we have seen also in the case of viral infections, that by reconstituting the gut microbiome, we also rebalance the immune system. Now we know that we have to do more. We have to reconstitute the brain microbiome inside the brain and the brain immune system. Now we have learned that the microbes reach the brain using immune cells as vehicles. We know that the macrophages travel through the lymphatic vessels, and therefore we hypothesize a new way of delivering our microbiome reconstituting product, the Bravo yogurt, in order to maximize the reconstitution of the brain microbiome. Now, here in these areas of your mouth and the throat, there is the highest concentration of macrophages in our body. It is called the Waldeyer tonsillar ring. And the macrophages from here, they go everywhere, including through the lymphatic system, they go to the brain. So here is where the macrophages are located, in these areas, and every time you have a deglutition, as you see, the bolus, in this case could be the yogurt, goes exactly where the macrophages are located. So as long as you keep the yogurt in your mouth and you swallow it slowly, you are stimulating the macrophages. The macrophages not only are activated and they activate the immune system, something that we have already seen, but they also pick up some of these microbes, not all of them of course, and some of these microbes are carried by the macrophages up to the brain. Of course, the macrophages have to be in good health, good strength, and efficiently stimulated. Therefore, we saw that the best way to deliver the Bravo is to divide the daily dose into 6 or more doses and keep the yogurt in the mouth as long as possible so that it continuously targets the cells of the lymphoid tissue that is associated with the mucosa. In this way, we also target the deep cervical nodes because, of course, we cannot use common methods to unclog these nodes, but we can decrease the inflammation, remembering that these areas of the mouth and the throat, they drain into the inferior and the superior deep cervical nodes. In other words, the same nodes where the lymph coming from the brain arrives. Now, this becomes very interesting, and I anticipate that the following results have not been published as yet, but I feel I have the moral duty to share them with you. Now, how do we demonstrate that we can reconstitute the brain microbiome? Of course, we could do a very simple experiment, take two subjects, one could be myself, one eats the yogurt, the other doesn't eat, and then we do an autopsy and we look for microbes in the brain. It's something, of course, can't be done, so let's avoid this type of experiment and let's go to petri dishes in vitro. Let's remember, if you go to Wikipedia or somewhere else and you look for cells of the central nervous system, you find neurons of all types and glial cells. Now we know that you have to add microbes. So microbes are cells of the brain, are cells of the central nervous system. They exerted their action. Now, here, what did we do? We do an experiment that at first, again, we must have been crazy even to think about this experiment. This is a petri dish. You can see the entire petri dish. These are cells, and in particular, these are human neurons. These cells are sitting on the petri dish. There is only their medium, no stimulus, nothing. What do? They sit there lazily. They try to connect to each other. This is a neurite connecting this neuron with this neuron. This is another one. Here you see another one, but they don't do very much. They simply sit there, they survive. Now, what we did, again, defies common sense. Cell biologists, of course, they try to stay away as much as they can from microbial infections, from microbes, because cells in a petri dish, they don't have an immune system to fight microbes. So if microbes happen to be in a petri dish, they quickly overcome the human cells. The human cells, they all die. So microbes are the enemies of the cell biologist. In this case, we took the Bravo yogurt, we filtered and diluted it, and then we simply did this crazy experiment to put the microbes of the Bravo yogurt together with the neurons. And when my wife first heard me saying this, she said, okay, you must be crazy because we kill all the cells, we ruin all the experiments, and this will be the result. No need to do the experiment. It's obvious that's the way it will end up. I said, well, let's do it with the Bravo yogurt. We have seen wonderful results in autistic children and other patients with neurological disease. At worst, we have killed a few neurons in culture, not a big deal. Look what happens when you add the microbes to the petri dish. Now, these very little tiny dots are the microbes. You see very many in the background. You see how many of these little dots. Look at what the neurons have done. They have formed a circuit. Look how many connections they are establishing. So here you see a major circuit, and then you see within the major circuit many short circuits. Now, this is the basis of neural network. Like networking in other areas of life, it is the conjunction of different subjects exchanging information. This is the basis of the functioning of our mind. And this was due to the effects of these tiny particles that are the microbes of the Bravo yogurt. Now, if at variance with the microbes of the Bravo yogurt, you put contaminants like E. coli or pathogenic microbes, then what happens is what my wife expected. All the cells died. So only certain types of microbes are able to elicit this response, that is to induce the neurons to form circuits, to form connections. The next slide is even nicer because here you can see many microbes. So these are different petri dishes. Of course, we always repeat the experiment. So you see now the neurons, how they are happy together, and they form several types of circuits. So you can see all these branches, one here, one here, one here, very long. So they exchange information. So again, we have the experimental evidence that if you put the right microbes together with the human neurons, you modify their behavior as the other scientists have written. Their influence is immense, and it is a very positive influence. Now, this is again an enlargement. This is taken from the web, and these are the microbes that you can see, like these little dots. These are the bifidobacteria, famous bifidobacteria. You can see them here. These are from the Petri dish. Of course, the focus is not excellent because we were focusing on neurons, not on the bacteria. Now, these results, I'm close to the end, are consistent with a recent study where we have elucidated and published the molecular mechanism that is at the basis of the evolution of the human brain, from the big apes to the Homo sapiens sapiens. Now, there is a gene that is almost unpronounceable. Its name is RANX2. Now, this gene is regulated by nutrients and probiotics. In other words, we and the apes have this gene. But the gene that is in humans is different from the gene that it is in apes, because certain nutrients, certain proteins, and probiotics, they are able to stimulate this gene. The gene that is in apes is slightly different and cannot be stimulated by proteins and probiotics. This is the paper that we published at the end of 2015, where we, the only important words are important implications for human brain evolution. Now, essentially, the size of the human brain had a so-called explosive increase in the past 10,000 or 20,000 years. So a very short time on the scale of evolution. So from the apes to the Homo erectus, the brain was small. In ourselves, the brain is much bigger. Why? Because the skull is much bigger. Why? Because this gene, RANX2, is stimulated by probiotics. In other words, when this type of hominid, or someone after this one, began to ferment food, and in other words had something that today we call probiotics, this gene was stimulated and the capacity of the skull increased, and it could accommodate a much larger brain. That is what we have today. And as a matter of fact, this is the paper demonstrating that the Lactobacillus rhamnosus, which is one of the components of the Bravo yogurt, activates this RANX2 gene. So these papers are all rather obscure. That's why they never received any public attention. But if we put all the pieces of the puzzle together, now everything begins to make sense. So in other words, we have demonstrated at the molecular level what had been hypothesized for years. That is, the explosive recent evolution of the human brain is or was due to changes in nutrition, and probiotics played a major role in this phenomenon. I was in Florida at the National Autism Conference last November, and I was among the public. This is a picture taken with my iPhone. And this is Dr. Rossignol, an autism expert, and who reported the clinical level of evidence of the different treatments that are proposed nowadays for autism on what is a strictly termed evidence-based medicine. And you may be interested in knowing that the first three are something of the first three of the treatments that, according to the principle of evidence-based medicine, are the most effective on the overall autism behavior are carnitine, probiotics, carnosine, sulforaphane. Now, carnitine and carnosine, they are proteins that are contained in milk and in fermented milk products like the Bravo. Probiotics, of course, they are contained in the Bravo, and the sulforaphane is a sulfated compound of the same class as the glycosaminoglycans like chondroitin sulfate. So with one single tool, we have the four most effective treatments for the overall autism behavior. Again, this is not my slide. This is a slide taken from Dr. Rossignol, who is not the one who wrote this slide, is the one who found all the collected evidence from the evidence-based medicine. Now, by combining these two studies, the one on the RANX2 and the paper published with Jeff Bradstreet, now we know why probiotics, or better, the fermented food products, are so important, not only in the evolution of the brain from the great apes to the Homo sapiens, but also in the development and function of the human brain, because the so-called probiotics, they are cells of the brain. And now we have found a way to deliver them to the brain to solve the problem of the inflammation of the deep cervical nodes, to stimulate the macrophages so that they can carry these cells to the brain. So now we have all the tools that we needed. And of course, as the advertising say, it is up to you to decide which type of microbes you want to have in your brain. I think this is the last slide, and now I am over for real, and I thank you for your attention.
Thank you so much. That was so great.
Thank you. Thank you for your time.
I know that we would have many, many questions, but like you said, you know, we'll have another time. Carolyn, was there anything that you wanted to ask or say before we let Dr. Ruggiero get like water or something and take off and have his dinner for the night?
I have some water.
I'm really excited to read some of the feedback and a lot of the questions and some of the buzz back and forth that Dr. Ruggiero can't see. So I'm even more excited now to plan and schedule another webinar as soon as possible. And again, we're just so incredibly grateful. This is revolutionary, and I so wholeheartedly believe it's going to change so many lives.
Yeah, and for people who want to order Bravo, the yogurt, can they go? Because I went on the other day and it said something about shipments being sent on January 15th. I didn't know if they could go on and order.
Well, you're asking me something that I'm not familiar with, because even though it is true that I hold shares in the companies, I'm working as a research rat, okay, or a rat lab. Okay. So I don't know these administrative details. I know that they're producing it. The Bravo yogurt is produced in Switzerland and in the USA. I know that in the next 2 weeks or so, it should be available again in the USA. They had a Christmas the backlog. I don't know what they did over Christmas, but so yes, it is available. It is fully available. The suppositories, however, they have to be ordered from Switzerland. And but you know, for this type of technical details, please send me an email, and I will forward the email to those who take care of these issues.
Okay, and I will say that I've used the yogurts. I'm using them, and I, before this, was making my own kefir, so I'm used to fermented foods, and it's relatively easy. There are instructions, specific instructions on how to make this, but it tastes amazing, and you know, I'm a strong believer in fermented foods and in, you know, enzyme-rich foods, and that was actually what brought me to discover you years ago. So Carolyn, you've used them, and you know, find them relatively easy to make, correct?
It seems intimidating when you first look at it, and it you know, looks really official and scary. But if I can do it, anybody can do it, and we're certainly happy to answer any questions and help with that end, obviously.
Okay, so I think because it's been so long, and I know that it's time for dinner for you and your beautiful wife, who also I hope that you can convey to her our appreciation for all of her work, and also for lending you to us for a while.
For a long
Don't
while.
worry. We are Italian, so we eat at about 8 p.m. So I still have 1 hour to go before dinner will be ready. Now, I really thank you, and I'm saying this from the bottom of my heart, because to have given me the opportunity to share this information with the people who may truly benefit from this information. It's really a precious gift. So I truly wish to thank you, and consider me at your service Whenever you wish to organize another webinar, you establish the topic. As I said, I have experience in these three fields: oncology, immunology, and neurosciences. So we can talk about oncology, or we can keep on talking about autism and other things. So these are my three fields of expertise, and I would be delighted to have another opportunity to talk with you and the beautiful audience that I cannot see. I only see the two of you. But I'm sure that all the people who are listening, if there is somebody still left listening, are beautiful people, and I'm very happy to have had this opportunity.
Well, the comments are that their lives are changed, that they love you, that they keep you in their prayers, and they are so grateful for everything that you've done. And just to confirm, you had said that, I think prior, that it was okay for us to share this recording.
I am begging you all to share this because I feel that my mission in life is not to publish papers, is not to make a little money out of these products. I feel that my mission in life is to help people. If this information can help people, please share it all. Don't be afraid. There is nothing that I have said that cannot be proved. Everything has been published in peer-reviewed papers. The only couple of things that have not been published, I would say, are rather irrelevant, but all the rest is published, is rock-solid science. So please, I beg you to share all this information. This is the greatest reward. You don't have to thank me, it's me. I have to thank you, in particular, if you share this.
Awesome. All right. Well, thank you so much, and we'll see you next time. We are now lifelong friends. Thank you so much.
Thank you. Thank you. Bye-bye.
All right.